All right, everyone. It's a beautiful Tuesday morning out there, and yet nonetheless, you're all in here with me. I'm not sure what's going on, but I'll do what I can to do some performance art for you here today to make it worth your while. About half of the class got an email from me in the last day or so suggesting that they hadn't looked at the first exam, the way I've been preaching and harping on you to do that. Remember, we've got these old tests on the pilot page for the course, and I keep telling you that's the best way to prepare for the exam coming up on September 29th. It looks as if many of the people who got that email had actually already looked at those first exams, and so the supplemental instructors and I and somebody at CTL are trying to figure out how the wires got crossed and all that. I'm expecting it's all going to be sorted out by Thursday, and what I've asked the supplemental instructors to do in particular is let's see if we can't get the pilot page of the course where you can see your grades reflecting whether you or not you got that 1 point of extra credit. The 1 point of extra credit, again, was for students who had looked at those first exams before midnight on Thursday of last week, right? And so, you know, we'll sort it out. Give us a little time here to figure out what's happened with all of that. And I'm, again, expecting that by Thursday of next week we'll have everything figured out, and you'll be able to see for yourself if you got that 1 point of extra credit or not just by looking at the pilot page for the course. More important than all of that, it's only 1 point. It's not that big a deal in the scope of 400 points for the whole class. More important than that, please remember that on September 29th, we'll have the first exam for this class. It'll be right here in this room for those of you who are here. For those of you who are listening to the Tegrity recording and doing the online version of the class, we've got a special room set up for you, a different room. But the best way to prepare for that test is look at some of those old exams. Boy, how I'm getting sick of hearing myself say this. But here's the thing that you should do. You should find 5 or 6 different versions of the first exam that I gave for this class on the pilot page. What you should do is check them out, get a sense for how it is I phrase questions. I've got punctuation issues. I've got wordiness issues that come into play. You know, unfortunately, we can't change me at this stage. I'm too old to learn some new tricks in terms of writing questions. And so I just need you guys to adapt to my idiosyncrasies in terms of how I phrase and ask questions. The best way to do that is look at some of those old tests. But then go beyond that. Don't just look at one of them. Look at 2 or 3 or 4 or 5 or 6. And what you'll see is that many of the questions look very similar from one year to the next. Figure out what's going on with those. That probably means that I think it's a particularly important concept. And even better, often you'll see that if I use the same question from one year to the next, the answer's a little bit different. And figure out what it is that changed in the question to cause one of the answers, a different answer, to be the correct answer. Because if you can figure that sort of stuff out, that means you get the concept. If I can change one of the parameters, one of the rules that are in play, and you can see how that affects the outcome of the scenario, you're good to go. That's the kind of mastery that I'm hoping you'll get for the things that I'm talking about. And the only way to really get you to do well in that sort of environment is to either take a couple tests, by which time it's too late for you to get a good grade in the class, or look at some of those old tests. So again, bottom line, look on pilot, find those exams. You know, I've been trying to incentivize you to just open one of them. I really hope you'll do more than just open them. I hope that you'll sort of wrap yourself up in them and really come to grips with what it is that we're talking about for this class. So anyway, enough said about that, except for one question. Yes?
When we print off the exams, unfortunately, the molecular formulas,
yeah,
the questions aren't showing up.
Right. So the comment here is that on the exams, you'll find that there are several questions for which somebody had to hand draw chemical structures. Things that got hand drawn on the exams were hand drawn on the exams. We didn't capture those electronically. In cleaning out my office last week, I found a copy of, I think it's last year's old test with those hand-drawn structures on it. And that one has been scanned and is on pilot now. So that gives you a sense of the kind of structures that I'll be hand drawing for you. For the ones for which we don't have those historical, Dead Sea Scroll kind of documents that were found, laying around in my office, I think that actually presents a nice opportunity for you, because you can look at the kind of structures that I drew in for that one exam and then just say, well, what kind of structures would I draw in? And use those as an example to fill in the gaps for some of the rest. Your drawing will be much better than mine, I'm sure of that. Okay. Good enough. Here's the deal. I am done talking with you about chemistry. Hooray, right? We're finally starting to get to the things here that are unequivocally biological, or at the very least biochemical. And you can see from the outline for today's lecture, I'm going to actually start talking with you about how some of these chemical pieces come together and make these smallest units of living things. We're going to start talking about cells, finally. Before I do that, though, I want to make sure that I take a sufficient amount of time to talk with you about those two biopolymers that I talked with you about on Thursday. I didn't spend too much time talking about DNA. That makes me feel sad, so let me fix that today. Before I get to the DNA, quick reminder, that's what a generic amino acid looks like. Okay, and I'm using this mostly just to illustrate something that comes up in the next couple of slides. But with amino acids, you've got that central alpha carbon. On one side, you'll have an amino group attached to that central alpha carbon. On the other side, you have a carboxyl group. There's always a hydrogen that's rounding out the valence for that alpha carbon. And then sort of the star of the show, the thing that's different from one amino acid to the next, is which of the 20 different R groups dangles off of that fourth covalent bond from that alpha carbon. So there are 20 different ones that you can dress up this generic amino acid with, ranging from something as simple as a hydrogen atom, in which case you're talking about an amino acid called glycine, to things with a couple of rings associated with them, like tyrosine and aspartic acid. When you link together one amino acid to another, remember this is polymers. These are biopolymers. This is polymerization. What you do is you take subunits and you string them together one by one to make a long chain. Linking them together involves dehydration reactions. A water molecule gets kicked out, and you make something called a peptide bond. All right, that's the covalent linkage between the one amino acid and the other amino acids that are already part of the chain. So there's a picture of a peptide bond for you. And again, this is just as a springboard to now remind you and go into a little more detail about what's up with nucleic acids. The monomers that get strung together to make the biopolymers that we call nucleic acids are called nucleotides. Okay? Nucleotides get strung together through dehydration reactions, just like all polymerizations take place through dehydration reactions. And the bond that gets made to link one nucleotide to the next in the chain is called a phosphodiester bond. You may have heard of that in some chemistry or biology classes in the past, but this here is a phosphodiester bond that's linking one nucleotide to another. So that's the linkage. Here are the nucleotides. There's a nucleotide. There's a nucleotide. And the bond that links one nucleotide to the next in the chain is a phosphodiester bond. All right, fancy talk. If you really want to know, ester is a bond that involves a carbon linked to an oxygen linked to another carbon. That's the organic chemistry definition of an ester bond. And it's a diester bond because there are two of those in these phosphodiester bonds, as well as a phosphate group, hence the phospho in phosphodiester. So bottom line, this is a biopolymer. The monomers that get strung together are nucleotides. The bond that holds the monomers together are phosphodiester bonds. That's kind of boring bookkeeping kind of stuff. Where I think it gets much more interesting, at least I see something that's much more interesting, is when we look a little more closely at the nucleotides themselves. Nucleotides have 3 component parts. All nucleotides will have at their core a 5-carbon sugar, sometimes referred to as a pentose sugar. Pentagram, 5-sided thing, right? So we've got a pentose sugar, a 5-carbon sugar. Ribose is what that sugar is. So you got a central sugar, and then onto that central sugar, you attach a phosphate group, right? That's the thing you see off to the left. And then the equivalent to the R group for the amino acids, the variable piece, because the sugar and the phosphate group are the same for all nucleotides, the variable piece, that third component, is what we refer to as a nitrogenous base. Okay, it's basic, acid base kind of basic, and it's got a nitrogen in it, hence nitrogenous base. There are only 5 different nitrogenous bases that we use in our bodies and living things on this planet use. There are only 5. And they come in 2 flavors. And I talked with you about this kind of quickly at the end of the lecture on Thursday. I told you one of those 2 flavors of nitrogenous bases are called pyrimidines, and the other flavor is called purine. Pyrimidines have the longer name and the simpler structure. They only have 1 ring associated with them. Purines have the shorter name, but 2 rings associated with those nitrogenous bases. Here's the deal. When biologists, particularly molecular biologists, get chatty about DNA, about nucleotides, nobody bothers to talk about cytosine. Nobody talks about thymine. Molecular biologists recognize the words, right? But they think you're putting on airs when you start talking about cytosine and thymine. Between friends, shorthand-wise, everybody just says C, T, U, A, and G. Right? The 1-letter abbreviation for those 5 different nitrogenous bases. In DNA, there are only 2 pyrimidines that get used. They are the T and the A. The T and the C, the cytosine and the thymine. And in RNA, instead of using T, U gets used. Okay? So that's the simple difference between RNA and DNA. There's one other difference, but we'll save that for later. And again, we've got these pyrimidines and we've got these purines. When DNA gets strung together to make these biopolymers, remember, you're effectively, for DNA, only using 4 different letters in your information storage alphabet. Proteins have 20 different R groups. Nucleotides, nitrogenous bases, are only effectively 4 different ones that you can string together. And you can store information if you've got a 4-letter alphabet, but a consequence of having so few letters is your words need to be a fair bit longer to convey some useful chunks of meaning. Okay? And so when we're talking about DNA, proteins are the things that get stuff done inside of cells. That's the biopolymer that's used to do stuff. But DNA is the molecule that we look to for information storage, and it's information that's stored simply in the linear sequence with which these nitrogenous bases get strung together on those nucleotides. The human genome is prodigious. It's huge. 3.2 billion nucleotides long. Okay, to spell out all the genetic instructions that you need, 3.2 billion G's, A's, T's, and C's strung together in a particular way that is effectively unique to you. Okay? So that's how much information it takes Again, that's an enormous amount of information. That's like CDs worth of information. It's like Encyclopedia Britannica sets of information. It's a massive amount of information. When these nucleotides get strung together, and I'll be talking with you about this in greater detail later in the course, but when they get strung together, we usually don't see DNA as a single strand. You've probably already heard many times that DNA is sort of famous for being a double-stranded molecule. The information on one strand is redundant with the information on the other strand. Let me show you a picture here that makes that case. So here's this sort of classic stereotypical image of what a DNA molecule looks like with the double strands sort of twisting around each other, sometimes referred to as a spiral staircase. If you take a closer look at what's happening within these twisted strands, what you'll see, this blue ribbon here is that phosphodiester backbone that I've been talking about. That's what's actually linking one nucleotide to another. Along this ribbon, there will be a phosphate group, a sugar, and another phosphate group, a sugar, a phosphate group, a sugar, all the way up, all the way down. But again, the interesting part, the key part of the molecule is which of the four different nitrogenous bases is also attached to those sugars, because that sequence is going to be important. And you can see here, you've got a purine with its bigger A, right? Notice how that's bigger here, because that's a purine. And over here, we've got a pyrimidine, a one-ring structure, a C. The G is another big one. It's a purine. It bulges out further. It should, well, kind of hidden behind the phosphodiester backbone here, but it's bigger. And notice, again, we've got two strands. On this strand, if we have a C, an A, C, and a G, on the other strand, we will have complementary to it a G, a T, a G, and a C. For every G on one strand, there's a C on the other. For every T on one strand, there's an A on the other. This is what we refer to as complementary or base pairing. So those nitrogenous bases are pairing with each other. From the picture, you can see that the way they've drawn it is A's have something pokey coming off of them, and T's have something kind of pointed to receive that pokey thing, right? There's a comfortable fit between a T and an A. Their charges can interact with each other. A positive charge on the A can nestle up against a negative charge on the T, and a negative charge on the A can nestle up against a positive charge on the A. It's a nice, good fit. And the same can be said then for the base pair that you see taking place between G and C, right? G's have a certain set of charges on their edge. C's have a certain set of charges on their edge, and they touch and they rub up against each other, and they fit nicely together. So that's base pairing. If you separate those two strands, all the information on the strand on the left will be effectively the same as what you get off the information on the right. They're kind of like negative copies of each other. But you could use the information from the one strand to make another copy of the other strand, and you can use the information on the other strand to make another copy of that first strand. Okay? And again, later in the course, we'll be talking a good deal more. When I get to really tell you about molecular biology, we can talk more about how cool that all is. So these are DNA molecules. Here's an example of an RNA molecule. We typically think of DNA as a double-stranded molecule and RNA as a single-stranded molecule. But RNA has nucleotides with nitrogenous bases, U instead of T, but otherwise all the same. And it turns out that RNA molecules can often fold up on themselves to make local stretches that are double-stranded and get these sort of funky, interesting shapes. Let me draw your attention to the fact that I said shapes. All right? In biology, structure and function are intimately related. The fact that RNA molecules fold up into a particular shape should telegraph to you that at some point, somebody, me, is going to be drawing your attention to the fact that that shape confers a particular functionality to the molecule. It's too early for me to get too deep into what those functions are, but notice RNA, because of its sequence of nucleotides, has a tendency to fold up and adopt a particular shape, and that shape will ultimately be responsible for it having a particular function. Enough said for now. Let me just tease you with that, and we'll get back to that in about 3 weeks, and we'll talk about how it is that the shapes of RNA molecules can help with catalyzing some reactions. But there we go. Now I feel I've done justice to the nucleic acids, and I can get to the topic at hand for the day, and that is, let's start talking about how all this stuff comes together, all these biopolymers, all the information storage, all the proteins that get stuff done, how that all comes together to give us cells, the smallest unit of living things. And let me really start by reminding you that this isn't the first time that I've kind of asked you about this. I'm sure you all remember before the class even really got underway, I gave you that pre-quiz, and one of those questions was specifically about one of the main points that I'll be making during the course of today's lecture. I asked effectively, what's the big deal, what's the difference between eukaryotic cells and prokaryotic cells? And the right answer for that question is C. And we'll talk about this quite a bit today, that prokaryotes are effectively defined by the absence of a feature. I'll tell you, historically, we know that that's a bad way to categorize things of any kind, particularly in living systems. Not having something is a bad reason to put you into a particular group. But it turns out that it works pretty well for this one particular instance. Not having internal membranes is what all prokaryotes have in common. And having internal membranes is something that all eukaryotes have in common. It's a pretty neat split between these two very large and well-populated domains of living things. Okay? There's another domain of living things, the archaebacteria. So 3 big domains, lots of diversity, lots of age, lots of evolution in all 3 of them. The archaebacteria and the prokaryotes have no internal membranes. Eukaryotes do have internal membranes. And again, that's what today's lecture is largely going to be about. Let me talk with you a bit about some of these other answers here. I think the people who chose A were just playing a game with me, right? Because this is the kind of answer. If I were to take a pre-quiz like this, I can tell you this now that you've taken it. But if I took a pre-quiz like this, I'd look for the most ridiculous answer I could find, and that's my choice, right? Because there's no points. I don't get a grade for this. OK, fine. A. There you go. Prokaryotes are carbon-based. Yeah. Eukaryotes are carbon-based too. We've been talking about that for a couple of weeks now. So I'm thinking most people realized that eukaryotes were carbon-based. Maybe there was a little bit of confusion on that, but everything, all living things that we know of, with the exception of the science fiction Huerta that we talked about a little bit, all things that we know that are alive, we know are carbon-based. But B, a fair number of people bid on that, suggesting that prokaryotes can photosynthesize, and they can. But it turns out eukaryotes can photosynthesize as well. That's what plants are all about. Plants are eukaryotes. They do a lot of photosynthesis, right? And so B falls flat on the second part of that answer. For D, prokaryotes have multiple chromosomes in each cell. Eukaryotes have just the one. That's kind of got things flipped around. I'll talk about that a little bit during the course of today's lecture. Eukaryotes typically will have multiple chromosomes. You and I have inside of all of our cells 46 chromosomes, 23 pairs of chromosomes. So we're eukaryotes. We've got a bunch of different chromosomes. Fruit flies have 8 chromosomes inside of all of their cells, 4 pairs of chromosomes. Bacteria, prokaryotes, have one. And I hesitate there a little bit because just this morning I changed the lecture notes to reflect the fact that that's not quite right. Many bacteria, it turns out, have 2 chromosomes. But back when I was taking freshman biology, the rule was prokaryotes had 1 chromosome, and it's just taken a while for the field of biology to recognize that some prokaryotes actually have 2 chromosomes. But yeah, the ones that we study the most, like E. coli, have 1, right? So prokaryotes, small number of chromosomes. Eukaryotes, bigger number of chromosomes. Not right for D for that question, because at the very least, eukaryotes never have just 1. I'm not aware of any eukaryote that has just 1 chromosome inside of their cells. So we've already got most of the people in the room, literally a small majority, have this stuff figured out. That's the big difference between prokaryotes and eukaryotes. And let me take a couple minutes here just to talk with you, to give you a sense of scale, give you a sense of perspective. It's actually in 2 weeks that you guys are going to be using some pretty expensive, fancy microscopes in the labs for this course. That's one of my favorite things to do. I love having the prep lab people go out to the Wright State Woods and come back with pond scum. And they literally do that. But you get to see so many bizarre things in the pond scum when you use the microscopes to look at it. It's like a whole other world, a whole other universe. I'm going to, just between us, I'm going to tell you guys a secret. See if the TAs pull this on you, because this is how I coach the teaching assistants. When you're looking at the pond scum and you see something really bizarre, right? It's like, what the heck is that, right? When you see something really bizarre, call the graduate teaching assistant over and say, what is this thing that's got like eyeballs on it? And what is that, right? And here's what I've coached them to say. It's like, I don't think I've ever seen that before. That's very interesting, good fun. Nobody has seen the stuff. There's like things that are living in the pond scum. 99% of the stuff, the bacteria living in dirt, nobody has ever named or photographed or characterized in any way. I'm not kidding. 99% of the bacteria living in dirt are unknown to science. You will probably be one of the first people, if not the first person, to see some of those things. Right? And so it's real easy for the graduate teaching assistants to come over and make you feel good about yourself and say, oh, I've never seen, that's very interesting. I've never seen anything like that before. But they can say that to everyone. So it's really cool. You're going to get to see some neat stuff in lab in 2 weeks. And you need microscopes to do it because of this scaling business that's going on. I'm a big guy. Right? I weigh a lot, more than I should, but I'm also about 2 meters tall, just to give you a frame of reference. Cut me in half, if you want to have a mental picture of what a meter looks like, right? And the cells that we're talking about here, like a human cell, are not 1,000th of a meter, but more like, let me make sure I get the right number here. They're not 1,000th of a meter. They're more like 1,000,000th of a meter, right? So 1,000th of 1,000th, right? Cells are tiny, and millimeters are at the upper edge of where you might start seeing some cells. So frog eggs, there's some really big outliers here, like chicken eggs, but frog eggs are a little bit bigger than a millimeter. A millimeter is 1,000th of a meter. Check this out. People with the very best eyesight that you can find can't resolve 2 points unless they're at least 1.5 millimeters apart. Okay? So what I'm saying in simple English is if you took a needle or a pin and you put 2 dots right next to each other, unless those 2 dots are at least 1.5 thousandths of a meter apart, even people with the very, very best of eyesight can't distinguish and say that that's 2 points as opposed to 1. If you want to see things at that kind of scale, like 1,000th of a meter, the only solution that we have is to enhance what we can do with our eyesight, and that's through microscopy. The light microscopes that you'll be working with in lab in 2 weeks are seriously top of the line. The people in the prep lab panic every year when they break them out because they're like $5,000, $6,000, $7,000 instruments, and they can be broken pretty easily. So, but this is top of the line equipment you'll be working with. But those light microscopes work really well, magnifying things about 1,000 to, if you really push them, about 1,500-fold. Now, things that are a millimeter in size become pretty easy to see when you magnify them to that degree. So 1,000-fold amplification or magnification is going to let you take a look at some things that you wouldn't normally be able to see. And then going further down the scale, to put things into some perspective here, this is the range at which light microscopes might be helpful. The smallest bacteria are actually too small to be seen even with a light microscope. And viruses are more like 1/10,000,000th of a meter in size, and they can't be seen with light microscopes either. So what's the smallest living thing is a question that I'll often get after class when I talk about this sort of thing. And I'll tell you, it's kind of debatable. We can go on for hours. People, I don't care to any longer, but people will go on for hours debating are viruses alive or not, right? If you accept that viruses are living things, then there's a gray area. But if you accept that viruses are living things, the smallest living things are on the order of 1/10 of a millionth of a meter in size, right? Super tiny stuff. And smaller still, you're getting into the range where you're talking about parts of cells. Later today, I'll be talking with you about ribosomes and some other proteins. You can't see those very effectively with a light microscope. You may be able to make out some hints of ribosomes in some of the cells you look at in 2 weeks. But we need an alternative to light microscopes for that sort of work, and those are electron microscopes. And we do have some electron microscopes at Wright State. They're like $100,000 instruments, and there's one that I know of, and nobody can touch that, so you won't play with that, but you'll see pictures from them. And then at the very end of the scale, when we're down into the billionths of a meter, we're starting to talk about the size of atoms. Right? So really tiny stuff. But again, where we started with this particular class. So just a rule of thumb, a nanometer, that N, we're talking about, when I talk about a nanogram or a nanometer, that's a billionth of a meter, right? 1,000th of a 1,000th of a 1,000th. When you're talking about micrometers, that is a millionth of a meter. And when you're talking about millimeters, that's 1,000th of a meter. Biologists throw those things around. Was that an assassination attempt? No, good. Okay, we're all okay. So when biologists throw these words around, we often don't stop to picture in our head just how tiny these things are. But a millimeter is 1,000th of a meter, a micrometer is a millionth of a meter, and a nanometer is a billionth of a meter. All right, so there's some scale for you. Let's get to work and talk about what's up excuse me, with prokaryotes and eukaryotes. I'm going to jump right into it. This is pretty much all that I'm going to use to talk about prokaryotes. That's a picture of a cartoon version and an electron microscope picture of a fairly typical looking prokaryotic organism. Remember, the deal is this. Prokaryotes and archaebacteria are defined by the fact that they do not have any internal membranes. Effectively, a prokaryotic cell, like the picture that you see up on the screen right now, a prokaryotic cell is effectively a bag full of goo. Right? Sometimes people chuckle when I say that. You guys are a much more, cerebral crowd. You don't think that's funny. But Wrap your head around that idea. A prokaryotic cell is a bag of goo, right? The bag is a plasma membrane. It's a phospholipid bilayer. I mentioned those in passing back a week ago when I was talking with you about the biopolymers that are lipids. I'm going to spend a lot of time talking with you about them a week from today when I get into the business of phospholipid bilayers and membrane functions. But the bag for this prokaryotic cell is a phospholipid bilayer, and inside of that bag, that boundary that defines, on the outside is the outside of the cell, on the inside is the inside of the cell, the goo. The goo has everything else that we need to have for a happy, functioning, working prokaryotic cell. Job number one, it's got to have some genetic information. So in that bag of goo has got to be some DNA, right? A chromosome or two. Where in the bag of goo? It doesn't matter. It can bop around. There's no constraints on where that chromosome is or hangs out. It just floats around, washing itself with the goo. And what else is in the goo? Well, there's a lot of water in the goo, right? But there are also going to be a lot of proteins that are encoded by the information on the chromosome. And there will be a lot of sugars and a lot of steroids and a lot of lipids, right? All the other types of biopolymers. But that's pretty much it. It's just a mishmash. There's no significant organization. It is a bag of goo. Okay? So that's it. And I'll tell you, when we talk about prokaryotic organisms, I have this tendency, I'm starting to appreciate that I do this, and maybe I'm playing this card too often, but when I say prokaryote, I hope what pops into your head is the word simple, okay? There's just not that much going on. And you'll really see what I mean when I show you a picture in the next slide of a eukaryotic organism. Because when I say prokaryote, I hope the first word, like a Rorschach test kind of thing, a word association sort of thing. When I say prokaryote, you say simple. When I say eukaryote, you say complex or complicated, right? Because you'll see when I show you that next slide of a eukaryotic cell that, oh, yeah, that's complicated, all right? So prokaryotes are really simple. They've got not a lot of stuff going on. There's a bag of goo. There's not a lot of organization aside from what you get from encapsulating the goo with a plasma membrane. Fair enough? And archaebacteria fit pretty much that same definition. No internal membranes, no organization inside, chromosomes just sort of bopping around, proteins in a mix doing their thing, carbohydrates there hanging out, doing stuff, all right? Prokaryotes and archaebacteria are simple. They're old. They've been around a long time. Best estimates today are that prokaryotes and archaebacteria arose at about the same time, about 3.8 billion years ago. The planet's only been a planet for a little over 4 billion years, so pretty early on, something happened that gave rise to prokaryotes and archaebacteria, and they've been chugging along ever since, and probably have not changed too substantially, at least in terms of their exterior appearance, for about 3.5 billion years. All right? Everything got sorted out pretty early. Basic structure was intact. Not much has been going on since. The design works. They can reproduce. They can make copies of themselves. Life is good. Nice, simple prokaryotes and archaebacteria. But something interesting happened from a biology perspective about 1.5 billion years ago. So about 2.3 billion years of life on the planet being nothing but prokaryotes and archaebacteria. And then something interesting happened about 1.5 billion years ago, and that is the origin of eukaryotes. So here's a picture of a typical or caricature of a typical eukaryotic cell, right? big step up in terms of complexity relative to what had come before with the prokaryotes and the archaebacteria. The big difference, the one thing you can really point to and say, that's huge, that's a huge difference, is eukaryotes have internal membrane-bound compartments. Okay? Prokaryotes and archaebacteria, no internal membranes. The only membranes associated with them are the ones that make the bag. For eukaryotes, though, not only do you have the plasma membrane that is the bag, right, but inside of the bag, you have additional smaller bags packed along. Internal membrane-bound compartments. And that was huge. The origin of something that could have these internal membrane-bound compartments about 1.5 billion years ago took life in a quantum leap kind of way beyond what had been playing out before. The bottom line, what really arises from those internal membrane-bound compartments is complexity. So that structure, what you're seeing here for this cartoon picture of an animal cell, there's a lot of stuff going on compared to what was happening with the prokaryote. That structure brings with it complexity, and the big payoff that eukaryotes get from having that complexity, I don't know, this is profound to me. It probably won't sound that important or interesting to you right now, but we can have, with those internal membrane-bound compartments, a wide variety of different environments inside of a cell. There's a compartment we'll talk about at the end of lecture today called a lysosome. Lysosomes are a membrane-bound compartment inside of eukaryotic cells. They've got a pH of 5. Inside of lysosomes, the pH is 5. Harken back to a couple lectures ago to what you've played with in lab here for this class. pH of 5, in a word, is acidic. You probably don't want to drink that, right? pH 5, that's pretty seriously acidic. It'll do a number on your hair, like change its color, maybe even have it fall out. Your skin would start to dissolve, feel kind of slimy. You don't want to mess around with pH 5. That's bad news. And yet I'm telling you, we've got a compartment inside of our cells that is cool and set up to handle that low pH. All right? The prokaryotes can't do that. They set up a part of the cell that's pH 5, and it's going to diffuse throughout the whole cell, and it's going to mess up everything inside of the cell. But eukaryotes, because we can compartmentalize, we can set up little pieces inside of our cells that are chemically very different from all the rest of the cell. And that stuff happens at pH 5 much differently than it happens at pH 7 or at pH 8. And we can play that game. If we need to have something special happen that happens best at pH 5, I got an answer for you. We'll send that thing that needs to happen and have it happen inside the lysosome. Prokaryotes don't have that luxury. They've got to do whatever it is they do to move that process along at neutral pH, and that may be 1,000-fold, it may be 10,000-fold slower a process, but we don't worry about that. You need pH 5. We got a compartment that can do that for you. You need a compartment that is specialized and amenable and set up to protect genetic information. We're good with that. We got such a compartment. We have these specialized compartments that do different things. Some have different pHs. Some have different chemicals. Some have different structures. Again, structure and function are so related in biology. And we can pull that off by having internal membrane-bound compartments. So, bottom line, the big difference between eukaryotes and prokaryotes and archaebacteria, internal membrane-bound compartments, that translates directly to complexity. And that complexity, the payoff for having that complicated internal structure is it lets us do stuff like build rocket ships and play pianos that prokaryotes can't even imagine. Even if they had brains, they couldn't imagine because they don't have the structure, the complexity that's necessary to pull that off. So complexity brings with it a lot of challenges, but it also brings with it some opportunities. When you look at this eukaryotic cell, what are you seeing? Lots of stuff going on. The big stuff that's going on is almost everything is a membrane-bound compartment, right? And during the course of the rest of today's lecture, I'll tell you some cute stories about each of those compartments. We'll start with the nucleus, we'll work our way out. We'll even say a little bit about things at the edges here, like mitochondria. But now, prokaryote, simple, right? Eukaryote, complicated. The big difference, internal membrane-bound compartments. This is an animal cell. This next slide shows you a plant cell, two different kinds of eukaryotic cells, still eukaryotes. We know it's a eukaryote because this plant cell has internal membrane-bound compartments. The bottom line is pretty much the same. There are some subtle differences. You'll see here these plant cells often have, in addition to the bag of membrane, they fortify it with a cell wall. Those cell walls are typically about 10 to 100 times thicker than the phospholipid bilayer that is the plasma membrane. There is an animal cell equivalent to a cell wall. Animal cells often have associated with their periphery something called a glycocalyx. It's a mess of sticky sugars that we dress up the outsides of our cells with. And they tend to be less rigid, but they're about as thick as these cell walls. You don't see these inside of animal cells. This is a picture of a chloroplast, right? That's where photosynthesis is taking place. We'll certainly be talking more about them. And then plant cells also often have these big sort of empty spaces, at least apparently empty spaces, called vacuoles. Sometimes those are used to store molecules like starches. Sometimes they're water. Sometimes they're just taking up space to give some extra bulk mass to the cell. But bottom line, eukaryotic cell. This is a plant cell. It's eukaryotic, complicated. Prokaryotic cell, simple. So prokaryotes versus eukaryotes, simple versus complex. You may think I'm giving prokaryotes a bad rap. We owe a great debt to prokaryotes, all right? Because of their simplicity, they are much easier to understand. They're much easier to break open and manipulate. Almost everything we understand about genetics and the central dogma of molecular biology and so many other things is rooted ultimately in the fact that we've been able to play tricks on prokaryotic cells. Right? So almost everything we talk about in this class is ultimately we know because somebody's beaten up on some poor prokaryotic cells at some point in the past and cracked them open and figured out what's going on. Because we share a common ancestor with prokaryotes and archaebacteria, the tricks that they use to get stuff done are the same tricks that we use. It's just because they're simpler, it's easier for us to manipulate and see what's going on. So they're very important from an experimental perspective. They get used in labs a lot. All right, so here's the plan. Let's work from the outside in. Let me spend a little time talking with you about that phospholipid bilayer, the plasma membrane, and then skip to the very center of the cell and start talking about the nucleus and work our way out to fill out the rest of the cell. I don't need to say too much more about prokaryotes at this point because they've got that plasma membrane and inside chemicals floating around, including DNA. So most of what I'll be saying now is going to be with eukaryotes in mind because we're going to be talking about what happens in those compartments. But all of those compartments and the plasma membrane have this cool structure, a phospholipid bilayer. This isn't the first time that I've shown you a phospholipid bilayer. A week from today, I'll spend even more time talking with you about phospholipid bilayers. But here's the deal. Lipids, these chains here of carbon to carbon and carbon to hydrogen bonds, typically 16 to 18 carbons long, are hydrophobic. They want nothing to do with water. Water wants nothing to do with them. there's sort of an exclusion zone, a détente that takes place between the water molecules and these lipid molecules. The lipid molecules say, we're going to hang out here. We won't mess with you. You hang out over there. You don't mess with us. We'll coexist, right? And so they set up their tails, they rub up against each other, and they set up this hydrophobic layer that's a water exclusion zone. And their negatively charged phosphate groups, because again, these are phospholipids, the negatively charged phosphate groups get oriented to the outside and the inside of this plasma membrane, because those negatively charged phosphate groups are something water does want to have an interaction with. And so they poke out to the watery environment on the outside and the watery environment on the inside. But in between, you've got a stretch of 30 or so carbons, the carbon and carbon to hydrogen bonds, that is very hydrophobic. And then within this plasma membrane, there will often be a variety of proteins present. And again, I'm going to spend more time talking with you a week from today about what those proteins do. But right now, let me plant this seed and we'll fertilize it and talk about it more later. What kind of amino acid R groups do you think are going to be on the surface of this protein, where the arrow is passing over it there? All right? What kind of R group? Because remember, there are 20 different R groups, right? I wanted you to pay attention to 4 different flavors of R groups. There are hydrophobic R groups, there are hydrophilic R groups, there are positively charged R groups, and negatively charged R groups. All right? I'm sure hoping you're being shy and not shouting it out, but those had better be hydrophobic R groups. All right? If they were hydrophilic or charged R groups, they've got no business being in this water exclusion zone that comes from those lipids there. What kind of amino acids are you going to use at this part of the protein? Right? In the linear sequence of the protein, as you're looking around, oh, we're at a part of the protein that's going to be poking out of the cell membrane into the watery environment out there. What kind of amino acids should those be? Well, I sure hope you're thinking hydrophilic at the very least, maybe positively or negatively charged, to really make sure they're there. Those proteins get anchored in place pretty snugly. They don't want to flip around, because if they start flipping, you start pushing those hydrophobic parts of the protein's surface up into the phosphate groups. Ooh, that doesn't feel good, right? And you start dragging the charged and the hydrophilic amino acids into the hydrophobic part of the bilayer, and that doesn't feel good. So these proteins get locked in. And again, structure translates to function. One more time, the linear sequence of the amino acids causes the protein to fold up in a particular way, to present certain things on their surface, right? And that in turn can result in a protein having a structure that causes it to be embedded in the membrane with a particular orientation. And then check out this particular protein. Look at that. It's got what looks like, it's sort of like a really long stretched out donut. Better, it's sort of like a short stubby straw, right? It's kind of hollow in the middle. What do you think might be happening there, right? That is a channel that could allow water-soluble things to come from outside of the cell to the inside of the cell, or go the other direction, right? So that's how it is we get water molecules and sugars that are water-soluble, for instance, from outside the cell to inside of the cell, and from inside the cell to outside of the cell. It's all structure, it's all function. More about that in a week. All right, so you got this membrane. For prokaryotes and archaebacteria, that's pretty much the whole story. You got a phospholipid bilayer, got some proteins embedded in it. Sometimes you dress up those proteins with cellulose to make a cell wall, or better, with sugars to make the cells kind of sticky. But that's pretty much the whole story. For eukaryotes, you've got that, but inside of that bag of goo, you'll have smaller bags of different kinds of goo. So, cell walls and membranes. Let's now just talk exclusively about eukaryotes and get to work talking about one of those internal membrane-bound compartments. Okay? Once again, eukaryotic cell, lots of stuff going on, lots of different membrane-bound compartments, conveniently shown here in a variety of different colors so that you can distinguish them. In life, they're all looking kind of the same. But let's start at the middle and work our way out. Let's focus our attention on the nucleus. There is, got that picture? Get rid of all the rest. There's that nucleus. Check it out. The nucleus, an internal membrane-bound compartment within eukaryotic cells. What kind of nuclei, that's the plural of nucleus, what kind of nuclei do prokaryotes have? None, right? Because prokaryotes don't have internal membrane-bound compartments. We're eukaryotes, we do. Among our internal membrane-bound compartments is the nucleus. Do you guys need me to tell you? I will, just for the sake of completeness, but what goes on in the nucleus is that's what stores our DNA. That's where our genetic instructions hang out. The nucleus is an internal membrane-bound compartment that is highly specialized to be a repository for safekeeping and accessing our genetic instructions as they're spelled out in our chromosomes, these long biopolymers of nucleic acids. Okay, so that's the primary function. The job number one for the nucleus is provide a safe place to store and to access the information in the organism's genome. Pretty much say no more. That's it. Okay? Now, as a tour guide, I'll tell you a couple other things that might be of interest to you about the nucleus. That's its job, is it stores information. In eukaryotes, that information, as I said, in eukaryotes, because it's not the case for prokaryotes or archaebacteria, but in eukaryotes, that information is stored in a structure called chromatin. Right? In prokaryotes and archaebacteria, it's just DNA. In eukaryotes, it's chromatin. The difference is chromatin is 2 parts protein for 1 part DNA, mass for mass. There's twice as much protein associated with our chromosomes in our nuclei as there is DNA associated with the chromosomes in our nuclei. It's a very intimate relationship. Pretty hard to tease the proteins away from that DNA. When I get to talking with you about gene expression, how genes get turned on and turned off, in about a month, that chromatin's going to come up again. It packages the DNA, it stores the DNA, it protects the DNA. It lets you pack more DNA into a small space than you would have been able to otherwise because of the organization that it brings. Okay? So in the nucleus, we're storing our genetic information. If you want to be really particular, you say that that information is being stored in chromatin, which is this association of proteins along with the DNA molecules. Also, I feel like a tour guide here, right? Also within the nucleus, you'll often, with a light microscope, you guys will probably see some of these in lab in a couple weeks. There's a darker spot that gets the name nucleolus. Okay, so it's a specific region inside of the nucleus. It is not membrane bound itself, right? It's just a region, a little sort of corner of the nucleus. It often shows up as darker. What's going on within the nucleolus is pretty simply this. That's where ribosomes get built. That's where a ribosome gets born. It's a ribosome assembly factory. Ribosomes are going to be coming up in the next couple of slides. The long and the short of it is that ribosomes are what translates the information stored in DNA into proteins, into that sequence of amino acids that gives rise to the polypeptides that are our proteins. Right? Those ribosomes get made, they get assembled in the nucleolus. Here are a couple of other features of interest for you for the nucleus. And I'll tell you just as an aside, I think it's curious that there seems to have been some sort of convention amongst textbook illustrators. For some reason that I don't understand, nuclei are always shown as purple, right? So Any book you open, you see something purple, they're probably trying to show you a picture of a nucleus. But some other features here. Check it out. There is the membrane that is defining this compartment that is the nucleus. If you look really closely, right? If you look closely at this picture, you'll see it, but if you look at this zoomed in version, you'll see it too. See those parallel lines running through there? You might want to look at that in the book or on the PowerPoint slides a little more carefully. Those parallel lines are supposed to represent to you a phospholipid bilayer. And I want to make sure you understand what I'm saying here. That parallel line, there's a line there and a line there, all right? Those parallel lines are a phospholipid bilayer. One line is the phosphate group on one side, the other line is the phosphate group on the other side. This phospholipid bilayer, the nuclear membrane, is kind of strange because it's not just a bilayer, it's a double bilayer, right? There's a phospholipid bilayer here, and there's a phospholipid bilayer here. The nucleus isn't just a membrane-bound compartment. The nucleus is a membrane-bound compartment with another membrane wrapped around the outside of it. It's, in grocery checkout terms, the nucleus got double-bagged. Cool? Drop the mic, walk away. I can't do better than that. That's the mental picture for you here. We got a bag of goo, which is the eukaryotic cell. Inside that bag, somebody put a double-bagged nucleus. One of the bags of goo has actually two phospholipid bilayers. It's a double bilayer. That causes, to draw attention to that fact, that causes microbiologists and some biologists to not refer to the nuclear membrane as a membrane, but rather as an envelope. Right? So a lot of times you'll hear people talk about the nuclear envelope. What they're referring to is that double bag that defines the boundary of the nucleus. Right? So it's double bag, double phospholipid bilayer. And what this picture is really supposed to be showing to you, this blow up, is that to get stuff in and out of the nucleus, you need to have something that spans that nuclear envelope, and those are called nuclear pores. That allows molecules like proteins, even big ones like ribosomes, to come in, to pass through in one direction or the other, as well as water and other smaller molecules like nucleotides. All right, so we got nuclear pores. You can see there is a zoomed in picture of the chromatin. The little blue line that's twisting around here is supposed to make you think of DNA. And see how it wraps around these proteins with their little tails poking out of them, and the tails can intertwine together to give rise to chromatin, and you get to see these sort of chromosome-like squiggles inside of the nucleus. So there you go. The nucleus specializes in storing information. You could think of it as the brains of the cell. It's the command center of the cell. I make the point in the lecture notes, I want to make sure I at least mention it to you here, that the thing that limits the size of eukaryotic cells, there's a practical constraint on how big our cells can be and still be effective in terms of keeping living processes going. The thing that primarily limits the size to which a eukaryotic cell can achieve is how long it takes for information to get from the edge of the cell to the nucleus. Because it's that flow of information into the nucleus, that information gets used to change the way that genes are turned on and turned off, to respond to changing conditions at the periphery and elsewhere within the cell. At a certain point in time, the edge of the cell gets to be too far away from that command center, and you just can't communicate what's happening at the edges quickly enough to the nucleus to be able to have an appropriate response in terms of a change in the way that genes are being expressed. So again, the practical limitation for the size of a eukaryotic cell is pretty much determined by how slowly diffusion allows information to get from the edges of the cell to the nucleus. Eukaryotic cells, on average, a typical eukaryotic cell is 10 to 100 times bigger than a prokaryotic cell. And that comes with a significant disadvantage in some sense for the eukaryote. We aren't as nimble. We can't respond as quickly to changes because in a prokaryotic cell, the DNA is right there in the thick of things, and it's small. For a eukaryotic cell, the DNA has to be able to get information to the DNA. It's got to travel a longer distance by a diffusion process, which is not a particularly fast process. That's what lab this week and last week were about. But also, it's got to fight its way through a double-layered membrane to get through, and that's another hurdle, another thing that slows down how quickly the information can get from outside the cell to inside the cell. All right, so you got a nucleus. Next out from the nucleus is another membrane-bound compartment. This one is called the endoplasmic reticulum. And they're showing you in this particular picture here just half of the endoplasmic reticulum. The next slide's going to show you the other half as well, but the half that you're seeing now is the half that's closest to the nucleus. That's the rough endoplasmic reticulum. And that's in contrast to the other half, which is the smooth endoplasmic reticulum. The thing that makes the rough endoplasmic reticulum rough and the smooth endoplasmic reticulum smooth is the rough endoplasmic reticulum with a microscope looks bumpy, rough, because embedded in the membrane are ribosomes. And again, ribosomes are what translates the information from nucleic acids into proteins. So they are the things that are stringing together the amino acids to make proteins. Some ribosomes are free-floating. They just hang out in the cytoplasm, stuff that's not membrane-bound within a eukaryotic cell. Other ribosomes, like these little bumps here, are membrane-bound ribosomes that are embedded within the membrane of the rough endoplasmic reticulum. So, what do you suppose is happening in the rough endoplasmic reticulum? Bottom line is what's happening in the rough ER is proteins are being made by those membrane-bound ribosomes. What kind of proteins? Somebody must be wondering. I'll tell you what kind of proteins. How about proteins that themselves need to be membrane-bound? Remember I showed you a phospholipid bilayer not too long ago with the short, pudgy straw? That was a membrane-bound protein. That protein doesn't do anything any good unless it's situated inside of a membrane. You know how you make proteins that need to be associated with membranes? You make them in the rough ER using those ribosomes that are embedded within that endoplasmic reticulum membrane. So that's one kind of protein. Membrane-bound proteins get made in the rough ER. Another kind of protein that gets made in the rough ER are the proteins that need to be exported to other membrane-bound compartments, especially also the proteins that need to be excreted from a cell. So we talked a little bit about insulin in this class. That's a protein, remember, specific string of amino acids that some horrible, nasty biochemistry professor made me memorize once, right? That's a protein. You know where that protein gets made? It gets made in the rough ER because that's what's going to let it get secreted from my pancreas cells out so it can be delivered out to the rest of my body. Right? So proteins that need to be secreted get made in the rough ER, and proteins that are membrane-bound get made in the rough ER, and proteins that need to go to other membrane-bound compartments, like the lysosomes, get made in the rough ER. I did say there was another kind of ribosome. They're the free-floating ones in the cytoplasm. They translate all the rest, which is pretty much all the proteins you need in the cytoplasm. They get translated by the ribosomes that are free-floating. One other small thing, putting on my tour guide hat. did you notice how the rough ER and the endoplasmic reticulum as a whole is really just a continuation of the nuclear envelope? This is a phospholipid bilayer here, running all the way around through there, but it's just a poking out of the same space that was the space that separates the internal bag of the nuclear envelope from the external bag of the nuclear envelope. So the inside of the endoplasmic reticulum is actually continuous with the inside, that space within the nuclear envelope. How about that? So, nucleus, attached in a couple spots to the rough endoplasmic reticulum, but the endoplasmic reticulum is sort of wrapped around these sort of stacks of sacs around the nucleus. Moving out a little bit further still, well, here's some pictures of ribosomes. Each of the spots there is a ribosome. But moving out a little bit further still, you get to the smooth endoplasmic reticulum. I'm not showing you a really good picture of the smooth ER. Maybe it's coming later. I see the Golgi is up ahead here. So you got your rough ER. At the periphery, there's fewer and fewer ribosomes. That's when we start talking about the smooth endoplasmic reticulum. Let me tell you what's happening in the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is not so much where protein synthesis is taking place, because the ribosomes are in the rough ER. Rather, the smooth ER is where lipid synthesis takes place. So you need to make more membrane. That's where it happens. Right? And you need to process things that are fat soluble if you need to have chemical reactions taking place to them. Say you need to detoxify a drug or a poison, right? Good chance that's going to happen within the smooth endoplasmic reticulum. You guys are probably not, for the most part, you're too young to be drinking alcohol, right? You know, but I'll tell you, someday when you get to be an old guy like me and you pound down a couple of beers every night, you know what's going on is that doesn't have the same effect on me today that it did back when I had my first beer. That first beer, wow, that felt real good for, you know, right away. Now I need a couple to get that same sort of happy feeling. You know what the difference is? The primary difference is that over years, I've conditioned my body to metabolize alcohol better, more efficiently than I had before I had been drinking alcohol. And that conditioning is largely the expansion of the smooth endoplasmic reticulum. That smooth ER is big and hefty in somebody who's accustomed to drinking a lot of alcohol. Somebody who's got an addiction to barbiturates would have the same sort of situation. Their smooth ER gets bigger so that they can more efficiently process those types of compounds, those fat-soluble types of compounds. So, smooth ER, what's going on there? You're making membranes, you're making, well, and you're doing things to metabolize fat-soluble molecules, things like barbiturates and steroids. All right. One other tour guide-y sort of thing, and this is my gift to you. I'm going to arm you now with a very powerful word that you can use to impress people of the opposite sex. All right, so next time you're talking about, you know, you're trying to size somebody up, and you're saying, try this. It's like, you have a fine reticuloendothelial system. All right, and they're going to accept. I'll give it to you a couple times. Reticuloendothelial system. All right. That's the word that hip biologists use when they want to talk about the complete package of the rough ER, the smooth ER, and the Golgi apparatus. All three of these combined together, that is the reticuloendothelial system. And I mention it partly because it's my gift to you. That'll help you tonight, maybe over the weekend, meet somebody that may become your significant other. Let me know how that romance works out for you. A couple of years from now, tell me about how your kids have cool reticuloendothelial systems too. But I mention it also because not only is it a cool word and it rolls off the tongue, you know, try it. You don't want to do it right now. Wait till you're in your car or something. Reticuloendothelial system. It's just got a sort of flow to it. It's like butter, right? It just sort of rolls off the tongue. But I mention it also because names like rough ER and smooth ER and Golgi apparatus are human conventions. We're sticking names on stuff that we see inside of cells. It's convenient for us to do that. It's convenient for us to say, oh, the rough ER looks a little bit different, and something different is happening there than in the smooth ER, and the Golgi apparatus looks different still, and something different still is happening over there. But it's a system. Within the cell, from a cell's perspective, there is no bright line that defines rough ER. Oh, we've switched over to smooth ER. They all just sort of blend together. It's a reticuloendothelial system. It's not like you have lots of ribosomes here and then you jump to no ribosomes in the smooth ER. It's a gradual sort of flow. And they're all interconnected. They're all kind of linked together as part of a single system. So you've got a rough ER. Moving further out, the ER starts to look smoother, and we'll call it the smooth ER. Moving further out, it gets more and more invaginated, more and more, like smaller clumps of the membranes, but still kind of connected together. And then we start calling it the Golgi apparatus. And so again, this is the end of the reticuloendothelial system. The beginning was the rough ER. You might say that the nuclear envelope was the beginning, but the end is clearly the Golgi apparatus. And by the time that we're out to the Golgi apparatus for this system, there's no longer any protein synthesis taking place. There's not even that much lipid metabolism taking place. What's happening in the Golgi apparatus more and more is that proteins are getting sorted so that they get into the little packages that get them delivered to the right places. They're getting glycosylated, so they're getting dressed up with special sugars that are being attached to them that help ensure that the proteins have the right function and also that they get delivered to the right final destination. And again, they're being bundled up in a way, packaged up in small vesicles so that those vesicles can be delivered to the appropriate final destination. It might be a lysosome, it might be outside of the cell, but little bundles of proteins wrapped up inside of an internal membrane compartment. And that's the Golgi apparatus. I got another question for you guys to contemplate between now and when we meet again on Thursday. And what I might want to do on Thursday is just have you guys talk amongst yourselves for a few minutes at the start of class. Kick around what you think the answer to this question is. So let me make sure you understand the question so you can kick it around intelligently. UTP, U here is short for uracil. Okay? It's one of the 5 nitrogenous bases. It's the one that gets us ultimately used to make RNA, right? So I'm talking here about uracil, triphosphate. Don't worry about the TP part just yet. But uracil, and I put a radioactive tag on it so it glows in some almost literal sense. And I'm asking you, after a few minutes, you feed a cell, a eukaryotic cell, that radioactive uracil. Where will you find the radioactivity? In which of these 4 different general parts of a cell might you find the radioactivity? We'll start with that at the beginning of class on Thursday, and then we'll finish the rest of the structures of cells and talk about how they do things. See you then.